Okay, in our last lecture video, we talked about the hyperbolic binding curve showing oxygen saturation of myoglobin. And now what we want to talk about is the binding curve for hemoglobin, because it turns out that it is not hyperbolic. So let's draw them on here. So here's the saturation of oxygen. So from 0 to 1. Down here on the x-axis is the pressure of O2. So we know that myoglobin looked something like this. So there's our hyperbolic curve. And we know that up here is the pressure of oxygen in the lungs. And down here is the pressure of oxygen in the tissues. And what we can see for myoglobin is that the total oxygen saturation of myoglobin doesn't change much between the lungs and the tissues. And that's good because myoglobin is for oxygen storage. You don't want it to change. You want it to hold on to it. However, if we instead look at the binding curve for hemoglobin, instead of being hyperbolic, it's instead sigmoidal. And so what we see here now is there is a big difference between the saturation level of hemoglobin in tissues and the saturation level in the lungs. And this is good too for hemoglobin's function, because hemoglobin's job is to pick up oxygen up here in the lungs, so get as saturated as possible, but then to release that oxygen, so decrease its saturation in the tissue, so the oxygen can be released and function as the final electron acceptor in respiration. And this is a pretty big change overall. So we're talking being about 96% saturated for hemoglobin in the lungs versus 64% saturation down here in the tissues. So big changes in binding under normal body changes for pressure of the oxygen within the body. And so what we can see for this is that the P50, so the pressure of oxygen at which hemoglobin is 50% saturated, is 26 torr. And you remember for myoglobin, the P50 of myoglobin, that's an M, was 2.8 torr. So what this is telling us is that the KD for hemoglobin is much greater than the KD for myoglobin. So remember, the KD is the dissociation constant. So this is telling us that hemoglobin has a lower affinity for oxygen. That means it needs more oxygen to occupy half of its binding site. So the KD is telling us, since the KD of hemoglobin is larger, there's more dissociation. So hemoglobin has a lower affinity for oxygen. So it takes more oxygen to occupy half the binding sites. So what this means physiologically is that myoglobin is going to bind oxygen under conditions where hemoglobin is going to release it. And this is going to form a really sophisticated oxygen delivery system from the blood to the muscles. And it works because the affinities of myoglobin and hemoglobin for oxygen are different. They have different KDs. And it's because the hemoglobin binding to oxygen, so that actual process that's giving rise to those KD values, is something called a cooperative process. And that's why the binding curve is sigmoidal in structure. So let's talk about what it means then to have cooperative binding. So we know that there are 4 hemes in one functional hemoglobin, because hemoglobin, remember, is a tetramer. And what it means is that when binding of oxygen to the first heme, so the first of the 4, that the binding of that first oxygen makes binding of the second, third, and fourth oxygen easier. That's the definition of cooperation. The binding of the first makes the binding of the remaining three much easier. And this is coming about because of a conformational change that takes place within the hemoglobin monomer upon the binding of ligands. So upon oxygen binding, there is a conformational change. So let's take a look at that. So hemoglobin has two different conformational states. One of them has a low oxygen affinity. So this one is referred to as the T state. You can think of that as the 10th state. And it's also the deoxy form. So we call that deoxygenated hemoglobin. So that's the structure of hemoglobin before any oxygen is bound. And they call it the 10th state because in between these different monomers here, we've got these ionic interactions. So we have a low oxygen affinity that's taking place right now. Upon binding, though, of that first oxygen molecule, hemoglobin, all four subunits undergo a conformational change to this R state. So this R state has a high oxygen affinity. It's also referred to as the relaxed state. So it's high oxygen, it's got oxygen bound, and this is oxygenated hemoglobin or oxyhemoglobin. So if we wanted to look at what the two states looked like in terms of binding curves, we could represent that. So here's our oxygen saturation, here's the pressure of O2. So you could think of the relaxed state, the high affinity state, as having a binding curve that looked like this, whereas the low affinity state, the T state, has a binding curve that looks more like this. So it has a low affinity, so it has a higher KD or a higher P50. And so hemoglobin, since it's a combination overall of both of these different conformations, one that has oxygen bound and one that doesn't, the net hemoglobin binding is the sum of the two, which is that sigmoidal curve. So that sigmoidal curve represents the transition then of hemoglobin from the T to the R conformation. So what's really cool is that when the oxygen binds to one of those monomers and that monomer undergoes its conformational change, that same conformational change is communicated across the four different subunits or the three remaining subunits of hemoglobin because of changes in the quaternary interactions between them. How that conformation change is communicated is unknown. So there's the idea is that what happens is when oxygen comes in and binds to the porphyrin ring, it binds to that iron center, that pulls the porphyrin ring into a different conformation, and the pulling of that porphyrin ring, so the pulling of the iron which pulls on the ring also pulls on this histidine residue. And that histidine residue is of course attached to the rest of the protein backbone. So you basically bind oxygen, pull on the iron, pull on the histidine, and then pull on the rest of the backbone. And that backbone then is connected, or that protein that the oxygen bound to is connected through ion pairs to the other subunits. So when you tug on that histidine, you tug on that protein, you also tug on the remaining three proteins as well. And there are two reported mechanisms for how this might happen. So one mechanism, so there's two possibilities. One is called a concerted mechanism. So in the concerted mechanism, all four subunits change at once. So let's say we're going to represent the T state as zeros or as circles and the R state as squares. So in a concerted mechanism, we're going to start with four monomers that are associated. They are deoxygenated. And then when one O2 comes in a concerted mechanism, they're all going to change at once. So now we have four squares. And that's going to allow oxygens to bind to all four of those subunits. The other possibility is a sequential mechanism. So in a sequential mechanism, one subunit changes at a time. So you'd have your 4 hemoglobin individual molecules in the tense conformation, and oxygen comes and binds to one of them and changes its conformation. And then it's going to happen one at a time to change the conformations until they are all in the relaxed state. So no one's figured out yet if it's a concerted or a sequential binding mechanism. So in our next lecture video, we'll look at how we describe then the binding of oxygen to hemoglobin in terms of its saturation relating to its KD, the same way we did with myoglobin.